The overall goal of this procedure is to establish three-dimensional organotypic cultures of the murine vestibular and auditory sensory organs.
This method can help answer the key questions in the inner ear research field, such as what are the roles of mechanical force and tissue stiffness during development of the inner ear sensory organs. The main advantage of this technique is that it largely preserves innate architecture of vestibule and cochlea and allows to study both molecular and mechanical signaling during inner ear development. Dr. Hudspeth and I first had the idea of this method when we realized that three-dimensional cultures can provide more physiologic conditions to study vestibular organ growth in vitro.
First, use 70% ethanol to clean and disinfect the working area and all the necessary dissection instruments, including No. 5 forceps and a hair knife. Then, use a sterile scalpel blade to make a longitudinal cut, split the head of the mouse into 2 halves, and extract the temporal bones. Transfer in 15 milliliters of HBSS buffer in a 60 millimeter Petri dish on ice. Next, use 2 pairs of fine No. 5 forceps to isolate the inner ears from the temporal bones. Then, transfer 3 to 4 ears at a time in a 60 millimeter Petri dish containing ice-cold HBSS buffer. Next, orient the ears with the medial side up to locate the utricle. Use the 2 pairs of No. 5 forceps to remove the cartilage.